In this video, we'll explore how scientists study populations in communities and ecosystems. You'll learn methods for estimating population size for both sessile and modal organisms, including random Quadrat sampling and the capture, mark, release, recapture technique using the Lincoln Index. Finally, we'll discuss how to test for an association between species using the chi-square test. These key concepts are some of the most important topics covered in the IB biology syllabus. Counting every individual in a population is often impractical, especially if the population is large, widely spread out, or difficult to access. Instead, ecologists use sampling techniques to estimate population size. These estimates are useful for tracking changes in population over time, making conservation decisions, and studying ecosystem health. However, sampling comes with sampling error, the difference between the estimated and actual population size. This is inevitable because only a subset of the population is measured. Bias in how populations are sampled, for example, only sampling near a water source, can increase the sampling error. The key to reducing bias is random or methodical sampling, which ensures each part of the habitat has an equal chance of being sampled. Quadrat sampling is commonly used for sessile organisms, like plants, corals, or barnacles that remain in one place and do not move. This involves placing a square frame, known as a quadrat, at random or systematic locations within the study area and counting the individuals within each frame. In order to ensure randomness, a measuring tape is placed at the edge of the habitat, and a random number generator determines which meter marker on the tape measure should be used. A second random number determines how many meters perpendicular to the mark the quadrat is placed. This process is repeated many times to randomly place the quadrats to count the number of organisms within the quadrat. The average number of individuals per quadrat is then multiplied by the total area to estimate the population size. The standard deviation of the mean number of individuals per quadrat provides information on the population's distribution, whether it's evenly spread or clumped in certain areas. If the standard deviation is high, then the population is clumped in certain areas, whereas a lower standard deviation indicates that the population is evenly distributed. For instance, in this example, we have two populations that we've estimated at 500 individuals. For both populations, the average number of individuals per quadrat is 5, and the square meters of the population area is 100, giving us the 500 population estimate. However, notice in this first population, the standard deviation is quite low, because every quadrat we collected data from, the number of individuals are either 4, 5, or 6. They deviate very little from the average of 5. This suggests little variability among quadrats and potentially an evenly distributed population. In this second example, we still have an average of 5 individuals per quadrat, but the standard deviation is quite high. We can see some quadrats have a high number of individuals like Quadrat 7 with 14 organisms, while other quadrats have no individuals present at all. So this indicates a population that is unevenly distributed in the environment. This could be because they're taking advantage of biotic or abiotic resources like access to water or sunlight, which are also unevenly distributed in the environment. This is an appropriate technique to estimate population size and distribution for sessile organisms, or organisms that remain in one place like plants. Modal organisms, like animals, require a different approach. For modal organisms, such as fish or mammals, ecologists often use the capture, mark, release, recapture method. This involves capturing a sample of individuals and marking them in a harmless and identifiable way. Capturing could involve a pit trap, like in this example, or a net in a marine environment, or other methods of capturing animals. Once captured, the animals are marked. The marked individuals are then released back into the environment and given time to mix with the population. After a set period of time, more individuals are recaptured in a second sample. While the capture, mark, recapture is a field method, the Lincoln Index is used to estimate population size with the formula population size equals m times n divided by r, where m is the number of individuals marked, n is the total number caught in the second sample, and R is the number of marked individuals recaptured. So for our example, M would equal 3 as we've caught 3 beetles in our pit trap, and this is the amount of beetles we've marked. When we re-release them, they mix with the population, and we take a second sample. In this case, N also equals 3, if we've caught 3 beetles in the pit trap on the second recapture. R is the number of beetles marked in the recapture, which in our example is 1. When we multiply 3 times 3 and divide by 1, we have an estimated population size of 9. This method relies on several assumptions, including that the marked individuals mix randomly with the population and that there's no immigration, emigration, births, or deaths between samples. Ecologists use various approaches to test for interspecific competition. Testing for interspecific competition means using data or experiments to determine whether two different species negatively affects each other's fitness because they share a limiting resource. Laboratory experiments can control variables and isolate specific interactions. For instance, determining the population growth of two species of paramecium grown apart versus grown together. Field observations, like seeing how barnacles settle into niches on a cliffside, as well as field manipulations, such as the removal of one species, can reveal the effect on the remaining species. If one species thrives in the absence of another, this suggests competition, though it does not confirm it without additional evidence. Field observations can be quantified using quadrats or transects that show patterns of species distribution and whether species are found together, apart, or have no association between them. To analyze the relationship between two species, the chi-square test can be applied to data from Quadrat sampling. By recording the presence or absence of each species across several sites, ecologists can determine whether their distributions are independent, that is, randomly distributed in the environment, or if they're associated, either they're found together or not found in the same areas. A negative association means they're rarely found together, and a positive association means they're often found together. Typically, many quadrats would be used in an ecosystem, and the number of quadrats that contained only one species, or only the other species, would be counted, as well as quadrats that contained both species or neither species. So, for instance, in this first quadrat, we can see that species B is present, but species A is absent. So we would mark that in the appropriate box in our data table here. In the next quadrat, the same is true. So we would add another count where species B is present and species A is absent. Many quadrats would be thrown, and the number of quadrats in each category would be counted and placed in a table. The total number of quadrats that contain both species, neither species, or either one of the species is our observed data. From our observed data, we can calculate an expected frequency. The expected frequency would be the number of quadrats in each category that we would expect if there were no associations between the species, and it uses the ratio of the two different species to determine a random distribution. This is calculated by adding up the row and column totals. So for instance, in our first row, 2 plus 12 is 14, and our first column, 2 plus 10 is 12. Then for each square, we would take that square's row and column total and multiply it, then divide it by the total number of quadrats thrown. For this first square, that would be 14 times 12 divided by 30, which equals 5.6. 5.6 is the expected value of the number of quadrats that should contain both species A and species B if the species are not associated. This is the null hypothesis, or H0, where the two species are distributed independently, meaning that they are not typically found together, nor are they typically found apart. They're randomly distributed in the ecosystem. The expected values are calculated for the other categories by multiplying each square's row and column totals and dividing them by the total number of quadrats thrown. You can check your work at this point, as the expected frequencies should add up to the total number of quadrats thrown, in our case 30. Now we can calculate the chi-square value. The chi-square value compares how different our observed data is from what the expected values of our model that says there's no association. The chi-square formula is the sum of the observed values minus the expected values squared divided by the expected value. We would do this for each category. So let's do this first one together. For both species being present, the observed value is 2 and the expected is 5.6. So when we plug that into our formula here, we get 2 minus 5.6 squared divided by 5.6. When we minus 2 by 5.6, we get negative 3.6. And when we square that, we get 12.96. We take 12.96 and divide it by 5.6 to get 2.32. We can now move on to our second category where species A is present and B is absent. And when we do the same calculation, we get 2.025. We do this for each category. We'll then sum these numbers together to get the chi-square value. So in our example, our chi-square value is 6.114. Once we have the chi-square value, we compare it to a chi-square table. To do this, we need to know the degrees of freedom in our dataset, which we calculate using the formula degrees of freedom equals m minus 1 times n minus 1. M is the number of rows in our data table, and n is the number of columns. Since we have 2 rows and 2 columns, our degrees of freedom is 1. We would use the row corresponding to 1 degree of freedom in the chi-square table. And in biology, we almost always use a p-value of 0.05. This table allows us to accommodate larger sets of data if we were comparing more sets of values. The p-value represents the probability that the sample we collected and the results that we calculated are due to chance, meaning they do not reflect what's happening in the real world. In the chi-square calculation, it's the threshold at which we decide whether the data we collected fits the model we use to calculate the expected values, which supports the null hypothesis that the two species are randomly associated. If the chi-square value is smaller than the value in the table for the given degrees of freedom and the p-value, this indicates that the observed data fits the model we use to estimate the expected value, and that the species are randomly distributed in the environment. If the chi-square value is larger than the value in the table for the given degrees of freedom and the p-value, it indicates that the difference between our observed data and the expected value, if there is no association between the species, is significant. This suggests that the two species are not distributed randomly. They could have a positive association, meaning they're often found together, or a negative association, meaning they are rarely found together. A significant result implies that the two species may be competing, cooperating, or influenced by a common environmental factor. In this video, we saw how ecologists can measure both population size and test for association between species. Here is a summary of the most important key concepts that we covered in this video.